Hi everyone and welcome to our core practical 10, which is investigating the effect of different factors on the rate of photosynthesis. Our objective in here is to understand how to measure the rate of photosynthesis by measuring the oxygen production and to investigate the effect of changing light intensity, light wavelength, temperature, and the availability of carbon dioxide on the rate of photosynthesis. For safety, remember to use eye protection. Avoid skin contact with the sodium hydrogen carbonate. Of course, any chemicals, you shouldn't touch any chemical and you have to wear your eye protection. Be careful not to raise or inhale any dust from it, of course. Don't handle electric sockets, plugs, or switches with wet hands. Wash your hands with soap and water, of course, normal hygiene. After all practical work is finished, and temperature, the availability of carbon dioxide, and the rate of photosynthesis. Yeah, forget this point. So, for the safety, I'll remember to use eye protection. Talk about your eyes, your mouth. Don't inhale. That's your nose, of course. Don't inhale or drink. Of course, you won't drink it, but you might soak it. Alright, don't do that. And your skin contact to be avoided with sodium hydrogen carbonate. So for the safety, I'm gonna look at myself and see what inside me could be changed or could be harmed. My eyes, use eye protection. My skin, avoid skin contact with the sodium hydrogen carbonate. And of course, be careful not to raise or inhale any dust. Alright, for the procedure, before starting this investigation, you should examine the equipment and read all the instructions carefully. Choose an independent variable. That is somehow our preliminary work. Alright, that's what you plan before carrying out the experiment, preliminary work. We're going to choose an independent variable, and I'm reminding you that the independent variable is the variable that changes. Or actually, let me write it in here, that the factor that changes. And the dependent variable is the result that's dependent on this, of course, on the independent variable. We gotta choose our independent variable, whether I'm gonna vary light intensity, wavelength, temperature, or the availability of carbon dioxide. And we're gonna record them, of course. That's what we're gonna discuss later on in solving. There are many other variables that should be controlled, but these have not been accounted for in the instructions. Identify all factors that should be controlled and give details of that, how they will be controlled. Plan it according to the limiting factor. So now these are the independents inside a list in here. And this was a gentle note. Since we only measure the rate of oxygen production, this means we are directly measuring the light-dependent stage. But of course, yet the products of the light-dependent stage are used in the light-independent stage, so it also affects the independent stage, which means by measuring the oxygen production, this means I'm measuring both stages of photosynthesis, but through direct measurement of one of them, which is of course the light-dependent stage. In practical work overall, I'd like my students to visualize the equipment. Like, as you can see, this is the setup, and the setup is one of the most important parts, because if you can understand, this means you can visualize. And our understanding helps us as well as memorization in such unit. So I'm gonna see that we have a pondweed in here that is placed inside. I'm gonna notice my pondweed in here. This is the plant that we have, and this plant is inside a funnel, okay, which is in somehow, we're gonna say all these terminology, but wait. First of all, we're gonna place a piece of pondweed about 10 centimeters long and place it in a large beaker of water. By placing the plant in an inverted funnel. So the first ever equipment I used in here is the inverted funnel. So just remember its name. But of course, in our questions, we're not going to write a list of the equipment. We're just going to include them inside a paragraph. Okay, I'm going to place it in an inverted funnel, which is in here the blue thing. Place it inside an inverted funnel with an upturned measuring cylinder. You can see in here we have an upturned measuring cylinder. It's not as much visible, but yeah, you can say it's in here, and that's an upturned measuring cylinder joined with an inverted funnel, and all these are inside that beaker of water. You can also call this photosynthesis with water. Make sure to remove any bubbles from pondweed by gently running a finger and a thumb over the surface of pondweed under the water. Is this going to be mentioned in the marking scheme? No. Cover one side of the beaker with aluminum foil so that light can only enter the beaker from one side. Yeah, this is one of the controlled variables. Add half a spatula of sodium hydrogen carbonate to water and leave for 5 minutes, so carbon dioxide found in excess is not to be a limiting factor. Okay, also know this one, yet it's not really a point in the marking scheme, so yeah, I don't suggest we write it. Add a spatula of sodium hydrogen carbonate. A spatula is like a spoon of that sodium hydrogen carbonate. Okay. Carry the experiment under 5 different light intensities. That's of course one of the most important points that I have to mention, my independent variable. And that's by changing the distance between the beaker of pondweed and the light source. And one of the most important points also is acclimatization. Acclimatization by allowing the pondweed to adjust to the experimental light intensity. Whenever I mention anything, like if I talked about the independent variable, I'm going to talk about the acclimatization because it's as important as everything else. Okay? Oh my god. Continuing, then we're gonna talk about our dependent variable, which is collect the oxygen gas produced by the upturned cylinder, bubbles pass into the capillary tubing, and use the syringe to draw any oxygen produced into the capillary tubing, and record the volume of the gas. This is very important. We're gonna collect the oxygen and record it in the syringe. Use the syringe to refill the capillary tubing, then begin recording again at another light intensity. And that's one of the uses of the syringe. It sometimes comes as a question, and write this down. Syringe is used to refill the capillary tubing for repeats. Syringe is used to refill the capillary tubing for repeats. Alright. Dependent variable. We said that already. But note that the dependent in here is not just oxygen. It's the rate of photosynthesis. So we're going to measure the rate of photosynthesis by measuring volume of oxygen gas. And of course, controlled variables. Controlled variables include temperature using a heat shield and the thermostatic water bath, pH using buffer carbon dioxide concentration by using the same concentration of sodium hydrogen carbonate, and monitor the temperature to make sure it's not changing by using a thermometer. All these were some points. Then repeat is also one of our main points. Repeat 3 more times at each light intensity and calculate the average mean value. Repeat 3 times at each of them and calculate the mean value. Furthermore, these are more examples to our thing of Core Practical 10. We talked now about the independent variable of light intensity. Now we have another independent variable, which is the light wavelength on the rate of photosynthesis. Keep the lamp at the same distance from the plant. Of course, now we're controlling it. Methodology: now use a colorless filter between the lamp and the beaker to allow white light color as a control experiment. Allow acclimatization for 5 minutes. A reminder for your physics IGCSE exam that light wavelength differs in every color of light. Light wavelength is going to differ. So from our equipment in here would be the colorless filter, is used between the lamp and the beaker. I have my lamp in here. Okay, and that is the beaker. I'm gonna add a colorless filter so that only white light can enter. Then I'm gonna repeat the experiment using filters of different colors. And that's how we changed the light wavelength. Secondly, we can also talk about the temperature, of course. Then we're gonna control the light intensity and the filter. We're gonna set up 5 thermostatic water baths at 5 different temperatures. Most likely it's gonna be 10, 20, 30, 40, 50. Okay. Plan an investigation to show the effect of carbon dioxide, of course, using sodium hydrogen carbonate of different concentrations. Furthermore, on this, we've got some limitations that might be asked on. It's actually not might, it's must be asked on, so you gotta memorize them and understand them, everybody. For the limitations, of course, we're producing our dependent variable, which is oxygen. This oxygen might be actually used into respiration, and that's actually a normal thing. Like, it's a gas. That same gas might be already internally used into respiration instead of being produced outside to be measured in a gas syringe. And some oxygen may not reach the gas syringe. This is the same thing. Not all gas collected is oxygen. Yeah, we measure oxygen, but we just know that some of the other gases may be different than oxygen, and this might cause a limitation. The light intensity may change when using different filters. The temperature of the water may change due to the heat from the lamps, and this is a good point. That's why we use a heat shield. Alright, so now a summary of our limitations, or like how to memorize the limitations and make them ready in your brain whenever you get asked this question. Now, if it asked me on the limitations of the rate of photosynthesis, I'm gonna have some points in my mind in order to answer this. Photosynthesis, what is my dependent variable? It's of course, measuring the rate of photosynthesis, which means I need oxygen. This oxygen might have some few problems with it. First of all, oxygen might be internally used in respiration. That's one point in your mind map. It might be internally used in respiration. Not all oxygen reaches the gas syringe. Not all of it can reach the gas syringe. And last but not least, point about oxygen, that not all gas collected is oxygen. Not all of it is it. Okay? These were some points. Apart from that dependent variable, I'm going to take a look on my controlled variables if they weren't controlled or if they caused some problems. Now, our main thing was light intensity. And of course, we control the light wavelength. Varying the light wavelength, if we are doing this, it might lead to a change in the light intensity. It's not constant now. So this is an error in our experiment. And last but not least, also we care about other controlled variables such as temperature. Now, temperature is affected by the lamp used. That this lamp will produce heat, which in turn increases the temperature. So this was my mind map when I talked about the rate of photosynthesis. I will mention 3 points in my brain to remember. The dependent variable, okay, the controlled variables, and their effects on one another. The dependent is, of course, the oxygen. The controlled is the temperature, and the effects, of course, like this one in here. After being asked the question to describe this core practical 10, you're gonna have a question of analysis of the results. Remember to convert your results to array in millimeters cube per minute and add these values to your results table. Calculate the mean results. This is a must. If you have values, you have to calculate the mean and include it in your table to add it to your results. Plot a graph to show the mean rate of photosynthesis for each value and indicate the spread of data using the range or standard deviation. Write a conclusion to discuss your results. Alright, so I'm gonna draw a table. I'm gonna plot a graph. And remember that the table has a mean value. And you're gonna get a conclusion. Of course, standard deviation, error bars. Then you're gonna say that you have to use like Spearman's rank correlation coefficient. Coefficient.